Morning, guys. Today we are going to look at how to set up an experiment to show that oxygen is actually the gas that is released during photosynthesis. Before that, let's do a recap session from our previous lessons. So we looked at the investigations on the necessity of carbon dioxide, and then after that we looked into how to carry out an experiment showing that chlorophyll is necessary for photosynthesis. Okay, so what we did for this was the first step for showing carbon dioxide is the gas needed for photosynthesis. You need to have 2 well-watered potted plants, and then you're going to place both of them in the dark for 2 days. This is to carry out destarching, which means to remove all of the starch that is made in the leaves. Because in total darkness, what happens is that photosynthesis will stop and the starch will be converted back to glucose. So this means that what you're going to find out is you're not going to have any starch present in the beginning of the experiment, because that is the result that you're looking for at the end of the experiment. So remember, the step 2 should be to check for starch. So you're going to pluck a leaf from both of the plants that you have destarched, and you're going to check that there is definitely no starch present. If there's no starch present, then you can finally carry out your experiment, which is from steps 3 to 5. So we're going to prepare jar A, which is to cover 1 pot with polyethylene bag. This just means like any plastic bag, and you're going to tightly cover it, and this is to prevent carbon dioxide from escaping the soil. Because in jar A, what we want to do is we want to make sure that there's no carbon dioxide inside this jar, because we want to test if carbon dioxide is necessary. So let's take it out. So how do we take it out? We're going to prevent any carbon dioxide escaping from the soil, because there are microorganisms here and maybe insects that can breathe out, that will respire, and then they will breathe out carbon dioxide. We don't want that. So we're going to cover it, and then we're also going to place it in a bell jar, and we're going to have something right here, which is a small beaker of sodium hydroxide. What sodium hydroxide does is that it's going to absorb and remove the carbon dioxide in the air in this jar. So what happens is we also need to trap the air here so that no air from the outside will bring in carbon dioxide into the potted plant in jar A. So this is called jar A. And then we're going to prepare jar B. So jar B is the control of the experiment. You're going to actually have carbon dioxide in here. So what you're going to do is you are going to cover this with a polyethylene bag as well, because you don't want the carbon dioxide will be coming from the soil, because you want to show that carbon dioxide in the air is the one that essentially is the one that's providing the bell jar with this bell jar and the plant with carbon dioxide. Well, eventually the carbon dioxide from the the soil will actually go to the air, but we want them both to be in a similar condition, so we want to be a bit more fair here. So we're gonna place that 1 pot into the diagram like this. So we're just going to not have any cork to prevent the air outside from entering, because we want carbon dioxide to enter here. This is called jar B. Then step 5, what we're going to do is we're going to get both of them out into the sun and expose it for about 8 hours so that it can carry out photosynthesis if it can, and then test it for starch after that. So what are the results that we're going to get? So in jar A, because there was no carbon dioxide, we're going to expect that it will remain yellow-brown. That means that no starch is present, no photosynthesis occurred, and the result for jar B is that you're going to get this like blue-black color. So from yellow brown to blue black color change, that shows that photosynthesis occurred because starch is present. Okay, now we're going to show chlorophyll is necessary for photosynthesis instead. So chlorophyll is a green colored pigment that you can find in the leaves in the chloroplast. And this chlorophyll, what it does is that it actually captures the light energy from the sun. And this is going to help to drive the conversion of carbon dioxide and water into glucose, and then essentially making oxygen as a byproduct. So step 1, destarch a well-potted variegated plant. Remember, variegated leaves are leaves that you can find on plants that have both areas that have chlorophyll and areas that don't. That's why they have kind of like whitish parts versus green parts. The green parts show that there is chlorophyll found in that leaf. So you're going to place it in the dark for 2 days to destarch. You're going to remove one leaf after the destarching and test that no starch is present in the beginning of the experiment. And then what you're going to do then is that you're going to expose that plant to strong sunlight. So in the beginning here, that there is definitely no starch. So no starch at the white parts and even the green parts because you destarched the plant. After destarching, you're going to expose the plant into strong sunlight for 8 hours, and then you're going to test for starch. So when you test for starch, okay, somehow the picture is in there. Your leaf is supposed to be in here. Remember to boil it to stop the enzymes from converting it back to glucose. Then you test with ethanol, and then you add ethanol to remove all of the green parts. The green parts, the chlorophyll, you're actually going to remove it because it's going to hinder you from seeing the color change better. So if you can remove the chlorophyll, the chloroplast will still be there, but it's just not going to be green. Then what you can do then is you're going to carry out the iodine test, and what's going to happen is that the parts that were green right here in the middle, they can carry out photosynthesis because they have chlorophyll. So they're going to have starch being made after 8 hours. So it's going to be positive for starch, whereas the whitish parts that don't have any chlorophyll, it's going to remain yellow-brown. This means that no starch is present even after 8 hours. That means that chlorophyll is definitely necessary for photosynthesis. So now we're actually going into this investigation about effective varying light intensity. You've done this in the previous lesson, which was to watch that video that was provided by Cambridge. And then also this is related to investigating carbon dioxide concentration, temperature, and how that affects the rate of photosynthesis, especially in aquatic plants. But when we talk about varying, it means that we need to change it. So if you have a lot of, for example, like intensity, you can see in that video that there were more bubbles being made. And today I'm just going to cover the fact that we need to know as well that oxygen is the gas that is released, because how do you know that those bubbles are actually oxygen, and why do we need them to be oxygen? Okay, so we're going to focus on the aquatic plants today and try to relate the rate of photosynthesis with oxygen released. So oxygen gas is actually released from plants as a waste product during photosynthesis. Waste means that they have excess of it, they don't really need it, but plants still do need the oxygen for respiration so that they can get energy to carry out other life processes. But because they carry out photosynthesis a lot, that means oxygen will be in excess, so oxygen gas will be released rather than used up during the daytime. The volume of oxygen released during photosynthesis can be represented by the number of oxygen bubbles that the plant releases. So this in turn can be used to approximate, means that you can assume that the rate of photosynthesis of the plant will be the number of bubbles being made. So why do we use aquatic plants for this? Because aquatic plants are easy, that you can easily see the bubbles coming from them when they are submerged underwater. And the same plant has to be used, because if you use different plants for the same experiment, even if it's the same type, even if it's the same species, the thing is that they will have different rates of photosynthesis. If you look at the equation for photosynthesis, you need carbon dioxide and water to carry out photosynthesis, but you also need light energy. And how do we get light energy? The amount of light energy that the plants will absorb depends on how much chlorophyll that they have in the leaves. Therefore, how much of this will result in how much of that. So if you have a lot of these things right here coming in, and also these things providing the energy, then you'll get a lot of glucose being made, hence starch will be produced. This will be stored as starch, and then you're also going to have a lot of oxygen gas produced. So remember, it's something like if you put a lot of stuff in, then you're going to get a lot of stuff coming out. And getting carbon dioxide depends on how much carbon dioxide is actually available, how much water is available, and also in terms of light energy, how much chlorophyll is available in that plant itself. So even though plants are of the same species, they have different genetics. They might not be identical fully. And so the amount of chlorophyll and chloroplast that each plant will have, even though it's the same species, will be different. That means that the rate of photosynthesis as well will actually change. So you need to use the same plant so that the amount of chlorophyll will be the same, the amount of chloroplast will be the same. So you're going to have the controlled part here that the rate will be assumed to be the same. You don't want to have different chlorophyll, and then you can't compare the results. Because you can easily control water. You can control how much volume of water you have. Carbon dioxide, you can do that as well. If it's aquatic, you can actually add stuff to the water so that it has this amount of carbon dioxide. So then if these parts are controlled, then you can actually measure oxygen and glucose and saying that is the rate of photosynthesis. So you have two types of plants. Types. So the first one is called hydrilla. This is a pond weed. That means that you can find this weed, this plant in ponds, and this is how they actually look like. Hydrilla. And then the other one is Elodia. They look pretty similar, but you can see that the shape of the leaves are a bit different. And this is Elodia. It's also a pond weed. You can find this in the pond. You can see in this picture clearly that there are little bubbles here. That is actually oxygen being produced. So to find out what gas is released during photosynthesis, what we need to do is we're going to obtain freshwater plant, for example, hydrilla or Elodia. And then we're going to place that water plant under an inverted filter funnel. Okay, so we're going to get a beaker. And then we're going to get the plant, the hydrilla or Elodia. Then we're going to have these blocks right here. This is to support the filter funnel so that it can stand. So, and then we're going to have a beaker of water. And then we're going to add like this test tube inverted on top. So this is going to help to collect the oxygen gas. Then we're going to fill it up with water, which is needed for photosynthesis. And to make sure that here no air is trapped in the beginning of the experiment, because actually at the end of the experiment there will be some bubbles here, air bubbles, but it's not air bubbles, it's actually oxygen gas bubbles. So what we're going to do to make sure that this plant has enough carbon dioxide is we're going to add sodium hydrogen carbonate. This is carbonate, and they can make carbon dioxide being dissolved in here. So dilute sodium hydrogen carbonate will be added here, so that carbon dioxide is enough for this plant to carry out photosynthesis. What else do we need? We have water, we have carbon dioxide. We also need sunlight for photosynthesis. So you can either put it under the sun, or you can place a light source near the plant for a few hours. And we also need to set up a control. What we can do to set up as a control is to prevent the other one, this plant from having photosynthesis occurring, therefore no oxygen gas. So what you can do is you can add that same plant either at the same time. So you can use a different plant, but like we said, different photosynthesis rate. But at the same time, you can have the same type of plant and then add it in darkness. Well, what we can do is that, the varying light intensity part, when we change the light intensity, when we want to change one factor, we need to use the same plant, just like in that video. But here, I think it's fine for you to set up a control, which is using a different plant of the same species, but you're going to place that in darkness for the whole experiment. So what you're going to notice is that over time, this aquatic plant is going to make bubbles, and those bubbles will rise up. And so that gas is going to displace the water downwards, because you have gas here, and gas is essentially air, not really air, but gas molecules is made up of matter, and they're going to take up space. So they're going to push the water molecules down, and they're going to have like an air pocket there. And that air pocket is actually oxygen gas being accumulated at the top of the test tube. So what we're going to do is that one thing you need to know here is about gravity, maybe, right? So gas is light, right? And so it's going to go on top here. So what we can do is that we can actually remove it, and make sure to remove it when it's half filled with gas. So what we're going to do is we're going to place the thumb over the mouth over here. So we're going to take it out, and then we're going to trap it. And you know that the gas will actually float up here. So it's totally fine. They're not going to go down here yet. Hopefully you're not going to get a lot of them escaping, because honestly, gas can carry out diffusion. So it can diffuse still out. But make sure that you have your thumb covering the test tube there. And what's going to happen is that you can actually invert that back up, and you're going to test the gas quickly with a glowing splint. And what's going to happen is that, so that's a glowing splint right there, not burning one, but it becomes a burning one because the glowing splint relights. And that shows you that oxygen gas is the gas that is produced during photosynthesis. So that is the end of that part. And the next part we're going to get into this week is to talk about rate of photosynthesis. We talked about this a little bit, and the rate of photosynthesis can be measured by counting the number of bubbles that this plant is making, and by the plant for a measured period of time. This means that we're going to count. So for a rate, when we talk about rate here, we're talking about how much is being made and how fast is being made. So when we're talking about how fast, we need to measure time. So for example, 3 minutes, they're going to count for the whole 3 minutes how many bubbles is being made. Like this, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And you're going to do that for 3 minutes. But remember that you need to do this in intervals as well. You need to repeat this maybe 3 to 4 times so you can get an average, because maybe the bubbles are too small and maybe too fast that you didn't count them properly. So you need to repeat this counting for over 3 minutes. So every 3 minutes for 4 times or 5 times, You're going to get that average so that you can be a bit more accurate with your results, and your results can be more reliable, because there will be errors, especially human errors in counting the bubbles. Make sure that you do that. Okay, so what's going to affect the rate of photosynthesis is light intensity, the amount of light available. So the amount of light available will actually determine how many bubbles being made, and you saw that being done in that experiment video. And also concentration of carbon dioxide. If you have more carbon dioxide, more bubbles can actually be made. And if the temperature is suitable, for example, if the temperature is too low, then you're actually going to see that the rate of photosynthesis and the rate of bubbles being made is lower. But if you increase the temperature to the optimum temperature, then it's going to make more bubbles. Availability of water, because water is one of the main, what do you call that? It's not a condition, but it is one of the factors needed. Oh gosh, I don't remember the name for it. Raw material, that's it. Yeah, water is a raw material, so you also need amount of water, and also amount of chlorophyll will actually affect the rate of photosynthesis. If this plant doesn't have a lot of chlorophyll as another of the same species, then the rate is going to be different. But that's why this part is actually difficult to control in an experiment, but we can control number 1 to number 4. But this part is pretty difficult. So all of these factors that affect rate of photosynthesis will be important for the next lesson, where we're going to learn about there's this graph, and it's about limiting factors. So we're going to see that next time. So what you can do now is you can stop this for today and try to answer the Go formative questions. So I'll see you guys next time. Have a good day, guys.